Welcome to Unacademy. This channel is Unacademy Future Doctors, and I am your educator for biochemistry, Dr. Shivangi Shukla. Continuing with our Image of the Day series, today is the second session. Tonight, rather, is the second session in the series. And let me first check whether the live stream is running smoothly or not. Okay, so my voice seems to be clear and the video is also clear, so let us continue. Okay, so we'll be discussing in this Image of the Day series. What we do is I put up an image before you guys, and I give you around 5 minutes to decipher, to infer the diagnosis, or to tell me the answer, what the image stands for, or to describe the image. Okay, so today is a descriptive image. Today you will need to describe what you are seeing, and that you have to do in the comments section. Okay, yesterday we had a diagnostic session where we associated two images together and we arrived at a diagnosis. That was phenylketonuria, and then we proceeded to read the theory part. Today I'll be putting up an image before you guys, and I think most of you, almost 99% of you, will identify that, and you will have to describe me. These kind of questions are asked in your professional examinations in the practical part. Okay, so the image based questions, very important, and you are given very less time. So I'm still giving you 5 minutes, but there you would be getting, in your practicals, you would be getting hardly 1 minute per image, or 1 minute is also quite a lot of time. Okay, anyway, so before we proceed with the session, I'll just quickly brief you about the Iconic and the Plus subscriptions on Unacademy for NEET PG. Okay, both of these subscriptions will provide you access to video lectures, live as well as recorded, from top educators across the country. You can also access their quizzes, the quizzes that they put up from time to time, which reminds me that there is an educator branded quiz for biochemistry that I have scheduled for you, that I have prepared for you guys, that is scheduled for 25th of February. All of you, please attempt that, and to enroll you have to put in the educator code DRSHUKLA, Dr. Shukla, and the enrollment is absolutely free, and there are attractive hampers from Amazon that you will be getting gift vouchers. So please do appear for that quiz. Okay. Now, Plus and the Iconic subscription will also provide you access to PDF notes, so that you don't waste time and you're not focused on just taking down notes. Rather, your sole focus lies on comprehension and understanding. And the Iconic subscription will, in addition, give you access to resources from Prep Ladder as well. Okay. And do not forget to put in the educator code DRSHUKLA to avail an additional 10% discount over and above the already subsidized prices. Okay, so this code DRSHUKLA, this is going to give you an additional 10% discount on either of the two subscriptions, Iconic as well as Plus. Okay, so while you are checking out, this is the last page where you have to make the payment. There, this is the column where you can apply the discount code. Okay, now, so this a very, very familiar image. Most of you, as I already discussed, almost 99% of you would be knowing what this image is of. So, first of all, tell me in the comment section, what does this image stand for? What is this image? And then you have to tell me, you have to describe this image. Okay, so I'll give you 5 full minutes to describe me what this image stands for, what is this image, and then describe this in 1 or 2 lines, as you would do in your practical station viva. Okay, what happens is there are a lot of stations there in the practical examination. What happens is you are given a sheet containing 4 or 5 questions, and you have to different, like one or suppose there are 5 stations. Okay, so what you will do, you will be divided into groups of 5, and you will be stationed at each station. Okay, all the 5 children will be stationed at each station, and they'll have to rotate. Okay, after every 1 minute. Okay, the buzzer will ring after every 1 minute, and they'll have to rotate. The person at station 1 will have to move to station 2, and so on. The cycle will go on till each and every student covers all the 5 stations. Okay, so it will be conducted in 5 rounds, and the invigilator will be ringing the bell, at which all of you will have to change positions. This interesting game, which appears to be a game, this is actually uh, a viva that happen, a practical test that you're subjected to in all your professional examinations. Okay, so try and answer this here right now. Okay, very good, Satish. This is TRNA. Now, describe this also, because this will not suffice. Okay, you will be asked for 1 minute. You will have 1 full minute for that. So you have to also describe this in 2 words. Okay, for 2 marks. Describe this as well. Half a mark you will get for naming it, then 1.5 marks you have for describing it. Describe this in 2 lines. How do you describe this? Look at the structure, look at the arms. First of all, what this structure is commonly called. So you can either be asked to name it, or write 2 important characteristics, or describe the structure, or write the function. As Satish has already written the function, very good. Very good, Sanjay. It is TRNA, yes. Describe the structure in 2 lines. Few important points about the structures. You can what can you write? You can write what is it the structure commonly called, and then each of the arms, just 1 line about the function of each of the arms. You can write the 4 important arms. Okay, wobble hypothesis also you're writing. All right. It does not work on the basis of wobble hypothesis, Satish. Though it is associated with the wobble hypothesis, but the principle, there's no basis of wobble hypothesis upon which it works. Mm-hmm. Last 2 minutes, any one of you can give your inputs regarding this. Come on, everyone, try and answer. Satish has given enough points for TRNA, but I'm still looking for the structure, which I'm going to discuss now. Okay, I'll discuss the structure of TRNA now. If there's anything that you guys wish to add, you can do that. Now, as is very clear from the comments, and it is very obvious that this is the structure of tRNA or transfer RNA. Okay, it is involved in protein synthesis by the process of translation. Okay, now what is the structure of tRNA called? The structure of tRNA is clover leaf structure. What is the structure? Clover leaf structure. Okay, clover leaf structure. All right, like a clover leaf. Okay, now it has four important arms and the fifth arm is the variable region, this one. So four important arms, we will see what are the four arms. Okay, now first of all, as an introduction for you to understand, tRNA is the adapter molecule. Now, what does the adapter do in our chargers whenever we go to a foreign location which does not work on the same voltage? The household voltage is not the same. Suppose you go to the US. Okay, there the appliances work at 110 volts rather than the 220 volts that India has, that our country has. So what happens is the electrical gadgets, they cannot function unless and until they have an adapter molecule that can uh, help help the appliances to function at 110 volts. So that is the adapter molecule that connects an appliance with the particular socket that works at 110 volts, even though they are not related to each other. This appliance is manufactured to work at 220 volts, but it cannot work at 110 volts, so it requires an adapter which can connect it together. Similarly, tRNA is an adapter molecule which connects the mRNA to the amino acid. mRNA and amino acids have no affinity to each other. Okay, but what is to be done? The genetic code that is present on the mRNA has to be translated into the amino acid. Okay, and various amino acids have to attach together to form the polypeptide chain. All right, now that is why to help amino acids get to help the mRNA translate into an amino acid, you need an adapter or a mediator molecule that will connect both of them, and this adapter molecule is tRNA, okay, or transfer RNA. Now, how does tRNA function? The first step is charging of this adapter molecule. Okay, how does charging occur? There is the enzyme amino acyl tRNA synthetase. Synthetase means it will require ATP. Synthetase, had it been synthase, it would not have required ATP. So synthetase means it will require ATP. Now what will happen? This amino acid is going to get attached to, this is the amino acid, it is going to get attached to the tRNA molecule with the help of the enzyme amino acyl tRNA synthetase. Okay. Now this enzyme and the adenosine and the AMP of the AMP present that is derived from ATP, it will form a complex. Okay. Now this intermediate complex will allow tRNA to bind with amino acid. Okay, and what will this be called? Amino acyl tRNA. Okay, and the enzyme and the AMP will be reutilized. Okay, so what is charging of tRNA? Attachment of amino acid to the tRNA with the help of the enzyme amino acyl tRNA synthetase and ATP. Okay, and this enzyme amino acyl tRNA synthetase is unique for each amino acid. Okay, it is unique for each amino acid. For every amino acid that is going to attach to tRNA, there will be a separate amino acyl tRNA synthetase. For methionine, there will be one amino acyl tRNA synthetase. For valine, there will be another amino acyl tRNA synthetase. Understood this much? So this is the first step where the tRNA molecule, the adapter molecule, gets charged with with the amino acid with the help of the enzyme amino acyl tRNA synthetase in the presence of ATP. Alright, now since we are discussing that amino acid is attaching to tRNA, let us discuss where it is attaching to tRNA. Okay, it is going to attach to the acceptor arm of the tRNA. Okay, now since we are discussing about the acceptor arm, let us quickly discuss about all the 4 arms. Now since we are already discussing about the acceptor arm, this is the acceptor arm on which the amino acid will attach with the help of the enzyme amino acyl tRNA synthetase. Now the other arm is D arm or dihydrouracil arm. Next is T-Cy-C arm or T-pseudouridine arm. And the fourth is the anticodon arm. The anticodon arm is complementary to the codon present on the mRNA. What is codon? Codon is the triplet code, 3-letter code for amino acid. Okay, this 3-letter code is present on the mRNA, and this the 3-letter code, the particular tRNA that is coming and attaching to the mRNA, this particular tRNA will have complementary nitrogenous bases on its anticodon arm, and this is how it will identify the mRNA. Okay, how it will be complementary to the anticodon region of the tRNA. Alright, so let us quickly see the image. See, this one is the acceptor arm. This one is the acceptor arm, where at the CCA terminal, what attaches? The amino acid attaches. Now, can you see, this is one polypeptide chain running from 5 prime, curling like this, and then finishing at the 3 prime location. So, this is one polypeptide chain that has folded extensively into a clover leaf structure. Okay, now this is the acceptor arm where the amino acid will attach. This is the D arm or dihydrouracil arm, and this is the T-Cy-C arm, and this is what? This is the anticodon arm. There is a 5th arm, that is the variable region. Understood? So this is how you can label. Always make sure you mark the 5 prime end and the 3 prime end. Very important. Understood? Can you see here you have the T-Cy-C group. So it is very easy to recognize this. Here you have multiple D dihydrouracil group. So it is the D arm. Okay. And this is the CCA terminal. 3 prime end CCA terminal where the amino acid attaches. Can you see this? This is the CCA terminal. Here the amino acid attaches. This is the acceptor arm. This is the extra arm or the variable arm. This is the D arm. This is the anticodon arm. And this one is the T-Cy-C arm. Understood? Now the tRNA has a clover leaf structure, as you can see. Now, what does the anticodon arm do? It is complementary to the codons present on the mRNA. Suppose the mRNA has UUU as the triplet codon. Then the tRNA containing AAA in the anticodon region will then recognize this mRNA and get associated with it. Then which amino acid will come and attach at the 3 prime end? The amino acid that is coded for by this codon, and this is what? Phenylalanine. UUU is phenylalanine. So this tRNA anticodon arm containing AAA is going to recognize UUU of the mRNA, and it is going to attract phenylalanine amino acid, which is coded for by UUU. So phenylalanine cannot directly go and attach to UUU because they don't have any affinity to each other. Now what do they need? They need a mediator, that is tRNA. Okay, understood this? This is how translation works. Now, the anticodon region, while much has been said about it, I have not described it yet. So let us describe the anticodon region, what this is. Okay, the anticodon region contains 7 nucleotides. 3 of them you would already know. 3 of them are the complementary nitrogenous bases that will recognize the particular triplet codon on the mRNA. What are the other 4? The other 4, if you read from 3' to 5', they are a variable base, a modified purine, then XYZ. This is the complement. These are the complementary bases to the codon. Then pyrimidine, then another pyrimidine. Understood? So these 1, 2, 3, 4, 5, 6 and 7 nucleotides. Understood? And why are you reading this from 3' to 5'? Because it is anti-parallel, complementary to mRNA. mRNA is read from 5' to 3'. And this tRNA, can you see the anticodon region? It is read from, this is the 3' end, this one. It is read from 3' to 5'. So the anticodon region is anti-parallel to the codon region present on the mRNA. Understood? The anticodon region, you will have a question saying the anticodon region consists of 3 nucleotides, and you will think that since there are triplet, the codon is a triplet codon, so the anticodon region will also contain 3 nucleotides. Okay, but it is not so. Okay, all right, understood? Next is the T-Cy-C arm or ribothymidine pseudouridine cytidine arm. Okay. Now, what does this T-Cy-C arm do? It is involved in binding of amino acyl tRNA to the ribosomal surface at the site of protein synthesis. What is the amino acyl tRNA? It is the tRNA containing the amino acid that is charged, isn't it? Did you see this here, this one? What is this? This is the amino acyl tRNA which is charged. Okay. Now, this amino acyl tRNA is going to associate with the ribosome. This is going to associate with the ribosome during initiation. How? The 80S ribosome is going to dissociate into 40S and 60S, and there will be a lot of initiation factors that will attach to 40S, and then there will be a ternary complex that gets attached to 40S. This ternary complex will be tRNA plus an initiation factor 2A plus GTP molecule. Okay, so this ternary complex that contains the tRNA molecule, this tRNA molecule is charged with the initiator codon. What is the initiator codon? That initiates translation. That is methionine, that is coded for by AUC. Okay, understood? So, this T-Cy-C arm is involved in binding of the tRNA through the ternary complex to the ribosomal surface of 40S ribosome at the site of protein synthesis. Okay, what does the D arm do? The D arm is involved in recognition of a particular tRNA species by its amino acyl tRNA synthetase during charging. Didn't I tell you that each amino acyl tRNA synthetase is unique for each amino acid that is attached to the tRNA? So, what is the structure that recognizes the particular amino acyl tRNA synthetase? It is the D arm of tRNA. So there is a tRNA molecule which contains the D arm, which will recognize this particular amino acyl tRNA synthetase and it will get charged. Now after getting charged, it will form a ternary complex with eukaryotic initiation factor 2A and GTP, and this ternary complex is going to attach to the 40S ribosome using the T-Cy-C arm. Okay, and already at the acceptor arm, it has the particular amino acid attached to it. And what is the anticodon arm? The anticodon arm will in turn read the mRNA. Okay, and how will it identify the mRNA? It will identify the mRNA through, how will it identify the mRNA? Through the Kozak consensus sequence. That is the initiator codon that is preceded at the 3' and 5' upstream region by purines. Understood? So, whenever it finds the initiator codon on the mRNA that is preceded upstream at the 3' position and the 5' position, at the 3rd and the 5th position upstream, it is preceded by a purine, then it is going to identify that region as the Kozak consensus sequence, and it is there from where the protein synthesis will start. Okay, understood? And Satish was talking about wobble hypothesis. Let us quickly discuss about wobble hypothesis also. Okay, Satish, so what is wobble hypothesis? I'll discuss with you. One thing is degeneracy of the genetic code, and the other thing is unambiguity of the genetic code. Okay, both of these are two sides of the same coin. What does it mean? Now you have 20 amino acids and you have 4 different nitrogenous bases or nucleotides. Okay, now suppose we have to form a genetic code in which one nucleotide or one nitrogenous base will code for one amino acid. So how many such codes will be there? 4 to the power 1 is equal to 4. So there will be just 4 codes, but you already have 20 amino acids, so you will not have codes for 16 amino acids. So you reject this idea. Now you prepare a code using 2 nucleotides, so it will form 16 codes. It is also insufficient. Now you prepare a triplet code using 3 nucleotides. Now it will form 64 codes, which is sufficient for 20 amino acids. Now out of these 64 codes, you have 3 stop codons, which when these stop codons are encountered, then translation stops. Okay, these are UAG, UAA and UGA. Okay, so you are left with 64 minus 3, 61 codons for 20 amino acids. Therefore, what this means is more than one codon can code for 1 amino acid. So, one amino acid can be coded for by more than one codon. Okay, but one codon cannot code for more than one amino acid. That means if there is UUU, it will definitely be phenylalanine. UUU cannot mean anything else than phenylalanine. But what happens is there can be other codes, other triplet codes for phenylalanine as well. Okay, Adarsh Kanoji is asking, when will the whole biochem be completed, ma'am? Adarsh, we are taking lectures for your comprehension. So we have completed we have completed the whole of metabolism. We have completed the whole of nucleic acids. A few miscellaneous topics like xenobiotics and hormones, digestion, these are left. Hormones also maximum part we have done. We are focusing now on answer writing and revision. Okay, and side by side we'll be taking up the miscellaneous topics as well. So do keep up with this. Alright. Anyway, so okay, so you understood this, the meaning of degeneracy. That means one amino acid can be coded for by more than one codon, but one codon cannot code for more than one amino acid. So this is degeneracy. This is degeneracy. And what is this? This is unambiguity. Degeneracy versus unambiguity. This one is unambiguity. Okay, got it. So this is now what happens is the degeneracy of the genetic code resides mostly in the last nucleotide of the triplet. Okay, that means the last nucleotide, that is UUU or UUC. Both of these will be the same. So can you see the degeneracy, that is one code, that is two different codons coding for one amino acid. This is mostly done by the last codon, by the last nucleotide. Okay. Now, therefore, the base pairing between this last nucleotide and the corresponding nucleotide of the anticodon is not done strictly by the Watson-Crick base pairing rule. What does this mean? The Watson-Crick base pairing rule is, it states that A will pair with T or U and C will pair with G, isn't it? But as you have seen that each tRNA and amino acyl tRNA synthetase is unique for each amino acid, okay? But one amino acid can have more than one codon, okay? But one tRNA can have only one anticodon, alright? Are you following this? So, what happens is that the last anticodon of the last nucleotide of the anticodon present in the tRNA can pair with another nucleotide present on the codon of the mRNA, and that need not necessarily be complementary. I'll give you an example for you to understand. Suppose this mRNA contains UUU and this is the tRNA molecule. Sorry, this is the tRNA molecule. This is the anticodon region containing AAA. So this particular tRNA it attaches to phenylalanine. Phenylalanine attaches to this. Okay. Now this is AAA. Okay, it contains the anticodon region AAA because it is attaching to phenylalanine. Now it will recognize this mRNA and it is going to attach phenylalanine to it. Suppose this mRNA was UUC. Now this is also phenylalanine. So the similar tRNA will come and recognize this because each tRNA is unique for every amino acid, because each amino acyl tRNA synthetase is unique for each amino acid. Okay. So now this AAA is going to combine with UUC. Now can you see C is attaching to A and these are not complementary. So Watson-Crick base pairing rule is getting violated here. So what does wobble hypothesis mean? It means that the third nucleotide can wobble or change its position. Understood? Understood this? So this is what happens here. Okay. Now each tRNA molecule contains a specific anticodon that is complementary to the codon on the mRNA. And for a given codon in the mRNA, only a single species of tRNA molecule possesses the particular anticodon. But some tRNA can utilize the anticodon to recognize more than one codon. Why more than one codon? Because that codon is wobbling at the last nucleotide because of degeneracy. So it stands for the same amino acid, so it will use the same tRNA, and in the bargain the tRNA is going to attach to a non-complementary nucleotide at the third position. Understood? For example, arginine, it is the degeneracy in the code is because it is coded for by AGA also and AGG also. Okay. Now the anticodon on the tRNA is what? It is UCU. Now suppose an mRNA with AGA comes in, then it will be a proper pairing U, C and U. But suppose an mRNA with AGG comes in, the same tRNA containing the same anticodon will come and attach to it. But what will it have? UCU. So it is UC and this one is U rather than it should be C, right? But because this is wobbling at the third nucleotide, A has changed to G and still it is coding for arginine only, so it will have the same anticodon and it will violate the Watson-Crick base pairing rule. This is very, very important. So Satish, have you understood? This is not the mechanism with which tRNA acts. It is one of the characteristics of the degeneracy of our code, of the genetic code. Okay? Understood this? So we'll conclude the session here. We'll meet again after a few minutes in the next session, that is mnemonic of the day. Okay, and we have two more sessions tonight at 10:45 and at 11:45 p.m. on the Unacademy app. That is the MCQ bonus biochemistry sessions. These are MCQ sessions that I conduct for revising the whole syllabus of biochemistry. So do attend those sessions also on the Unacademy app from 10:45 to 11:45 and 11:45 to 12 or 12:45. All right, then see you guys.